Welcome to the YouTube live class for class 12th Botany. Once again, I am Dr. S.S. SARDAR from RDMS HSS, welcoming you all to another new exciting and interesting topic, and that is none other than your DNA replication. Now, this is a very much awaited topic and this is a very exciting topic because DNA is considered, or DNA is the molecule of life, because if DNA molecule is there, now it has the capability to inherit itself from generation to generation, and then it is also being able to express itself finally to the level of protein through mRNA. So, DNA is the basic theme of our life, or it can be called as the biomolecule of life. So, we should know that how the DNA replicates and at what time and how it replicates and what are the mechanism that lies behind the DNA replication. So, let us have a look on that. So, what is DNA replication? So, it is nothing but copying the molecule of life. Okay? So, it is the molecule of life, as we know that whatever we may learn, whatever we may study. Now, this is according to that what, how, whether it is genetics, molecular biology or whatever. Now, DNA is very important biomolecule and it has a capability to synthesize all sort of molecules by giving the label of gene expression, because gene is nothing but a segment of DNA. So, the whole DNA content of a cell, it has to be duplicated or replicated, or it must be synthesized to double its content before undergoing a cycle of cell division. So, what is DNA replication? It is a process by which DNA makes a copy of itself during cell division. Once again, replication or DNA replication is the process, it is a process by which DNA makes a copy of itself during cell division, and this happens during cell division. As we know, now the cell division process is divided into an interphase where the cell prepares itself for undergoing a division phase and a dividing phase. So, within the interphase, now when the cell prepares itself to undergo a division, at this particular point of time, now it increases its nucleocytoplasmic ratio. So, this kind of preparation is under three phases. One is G1 phase, the other one is S phase, and the final one is G2 phase. Now, within this G1, S and G2 phase, now the S phase is crucial as your DNA replication happens during S phase of cell division. Okay? So, in replication, DNA makes its own copy. Now, let us suppose if this is a DNA molecule, now this DNA molecule will it will prepare another molecule of this or another molecule like this. So, instead of getting one pen, now you will get two pen of equal nature and of equal size, shape and everything. Okay. So, it will be exact replica of the old one. So, how it happens or what is the mechanism behind that, that we will see today. So, in molecular biology, DNA replication is the biological process of producing two identical copies of DNA from one original DNA molecule. As I said you, now if this is a pen, now it will produce another pen exactly like this. So, it is a replica or it is a replica of the original DNA copy. So, DNA replication occurs in all living organisms, acting as the most essential part for biological inheritance, because now you see, now when meiosis happens, okay, now what happened? Now, if we have 2n number of chromosomes in a cell, okay, now what will be the chromosome number of the gamete? It will be haploid in nature, obviously. So, if the DNA content would not be doubled before the division. Now, in each cell division, it will be reduced to half and half and half. So, it will lead to its non-survival. Okay. So, that is why DNA replication is very important. Now, let us have a look. What are the features of DNA replication? Now, exactly what DNA replication is, let me tell you what it is. Now, DNA replication. Otherwise, we may call it as DNA replication or duplication or DNA synthesis. Now, when it happens, what is the time when it happens? It happens during the S phase of cell division. Now, S phase remains in the interphase. Now, here what happened? The content, the DNA content is doubled. If we have X, then it will be doubled to 2X. Okay. Now, how it replicates? Now, let us suppose I have a parent DNA molecule like this. As we know that DNAs are helical, it is a double helical structure. Now, as it has, okay. Now, if this is the DNA molecule, now it will replicate into 2 DNA molecules like this. So, after replication, so as it will produce a replica of itself, okay. So, after replication, now this will be like so instead of getting 1, now we are going to get, wow, okay. So, instead of 1, we are going to get 2 identical DNA molecules, okay. So, what we will call it as 2 identical DNA molecules, okay, which will be an exact replica of this one. If this is the original one, this too will be the exact replica, okay. But one more thing, that is that okay, one thing is that now in this, now if I will tell you that this is the DNA molecule, now what happened during replication, right? This is the DNA molecule. Now first of all, these 2 strands, now as you can see there are 2 strands, one is this one, the other one is this one, okay. These 2 strands, they separate from each other, forming 2 templates. Now here these are called as templates, okay. So, 2 templates will be formed by dissolution of the hydrogen bonds which are present between them. So, how the double helical structure is maintained? It is maintained because of the hydrogen bond, and this hydrogen bond will first of all, it will what? This will first of all this will dissolute or this will break and it will create two single stranded template. Okay. Basing upon this two single stranded template, another two strands will be created. Okay. So, here basing upon this template strand, another new strand will be synthesized. Okay. And basing upon this template strand again, another new strand will be synthesized. Now, you are getting what? You are getting from one DNA molecule, you are getting two DNA molecule, but here one strand is old, the other strand is new. The same is for this one also. One strand is old, the other strand is new. So, the new strands are synthesized, whereas the old strands or old single strands, they act as template for the synthesis of new strand. So, what this nature of DNA replication is called as, this is called as semi-conservative nature of DNA replication. So, this is the nature that is semi-conservative. That means, out of these two strands or out of these two DNA molecules, now here one strand is old, this one is the old strand and this one is the new strand. Similarly, here one is old, the other one is new, and this nature is known as semi-conservative nature of DNA replication. So, this is the basic theme of DNA replication, where you can see that how a single DNA molecule is capable of producing two DNA molecules. Now, next, so as we are discussing about what are the characteristic feature of DNA replication. So, first of all, this is semi-conservative as we have discussed. Now, what is semi-conservative? Now, here if we are going for what? A DNA replication, what? This is a molecule. So, it will what? It will separate from each other, making two templates, and next another molecule will be synthesized over this, and this will be two strands of two DNA molecules, okay, of similar types, but here one strand is what? Old, the other strand is new. One strand is old, the other strand is new. So, this is called as the semi-conservative nature of DNA replication. Okay. Now, next is that it is semi-discontinuous. Okay. This is semi-discontinuous. So, what do you mean by semi-discontinuous? Now, here out of the two strands, let us suppose we have these two strands. Okay. Now, here when they will generate a template, now out of this, now a template is created like this. Now, one strand, it's synthesized in a continuous manner, okay. The other strand is synthesized in a discontinuous manner like this, okay. That we will discuss more in details. For example, you can see so for example, we can see yes. Now, the semi-discontinuous nature is says that, now you see this is one template, this is another template, this blue color one. Now, you see one strand is synthesized continuously called as leading strand, okay. the other strand is synthesized discontinuously, okay, and in terms of fragments. It is not continuous, okay. In terms of fragments, they are synthesized and the fragments are called as Okazaki fragment, and this strand which is synthesized in fragments, in parts, now they are called as what? Lagging strand, okay. So, this nature is semi-discontinuous. What it means? Now, one strand is synthesized continuously, the other strand is synthesized discontinuously in fragments, okay. So, this is called as the semi-discontinuous nature. So, this is called as the semi-discontinuous nature of DNA replication, okay. So, one is semi-conservative, the first nature is semi-conservative, the second one is semi-discontinuous, then the most important, the synthesis occurs from 5 prime to 3 prime direction, okay. Now, the synthesis occurs from the 5 prime direction to the 3 prime direction. Let us suppose this is the strand of DNA. Now, if this is 5 prime to 3 prime, the other strand will be synthesized, okay. Now, the other strand that has to be synthesized, it is from the 5 prime direction to 3 prime direction. So, this is the direction that it has to maintain. So, always the DNA replication, it proceeds from 5 prime direction to 3 prime direction, okay. Now, next it takes place once per a single cell cycle, as you know. Now, that as I told you that during the S phase only the DNA synthesis occurs, it doubles the content of DNA, after which now you will get the duplicated DNA content, and this content will be halved and it will be this will be given to 2 daughter cells in equal amount. So, that is why DNA replication happens once during a single cell cycle. Now, next is replication happens in single replication unit or replicon. So, what is replicon? Replicon is the unit of replication. So, in simple term, replicon is a unit of replication. Now, for example, let us suppose we have a very long molecule of DNA, okay. Now, it does not mean that this will be synthesized, they will be converted into 2 templates and they will be synthesized what from this end to this end like this. There may be certain points, okay. There may be certain points from which now this will constitute a replicon or a replicating unit. So, replication happens in single replicating unit, or this is called as what? Replicon. Now, what is replicon? Let us have a look. Okay. So, let us have a look on what is replicon. So, before going to replicon, let us have a look on what are the tools that are required for DNA replication. Now, tools means several. DNA replication is a very complex process, it is a very elaborative process. So, lots of proteins and enzymes are involved in DNA replication. So, some of the important or very important enzymes and units are, or the proteins are the DNA polymerase, DNA ligase, helicase, topoisomerase or DNA gyrase, RNA primase and single stranded binding protein. So, these are the tools basing upon which we will study the DNA replication process, and all of them togetherly comprise of the replisome. So, what is a replisome? Replisome is a DNA replicating system. That means, this is a system where these molecules are involved in fulfilling or in accomplishing the replication process. That means once how the DNA synthesis will start and where it has to terminate, everything is regulated or everything is controlled, and it is being done by different units or by different enzymes, proteins and all, and togetherly they form the replisome. Okay. So, here I can say that it takes place, or DNA replication takes place with the help of DNA replicating system known as replisome. So, what are the units or what are the different enzymes and proteins that are being involved in the replisomes are DNA polymerase, DNA ligase, helicase, topoisomerase, RNA primase and single stranded binding proteins. Okay. Now, before going to, now let us have a look what is a replicon. Okay. So, what is a replicon? Now, a replicon is a unit of replication. Okay. Now, the prokaryotes have a single replicon. The prokaryotes have a single replicon. What it means? Now, as you know, the prokaryotes have a single circular double stranded DNA. Okay. Now, it has a single point. I will tell you what it is. Now, let us suppose this is a point. Okay. So, from this point, now your DNA replication will start. Okay. And it will go on ending off, or it will proceed to end off or in creating two double stranded molecules. Okay. So, it will go on ending off with creating two double stranded, two such molecules. Okay. When once it is initiated means it goes on replicating this whole DNA length. Okay. So, once it is being initiated from this point, what this point is called as? This point is called as origin of replication. So, from this origin of replication, this will go on synthesizing the total length of DNA and it will give rise to two DNAs like this. Okay. So, in prokaryotes there is a single replicon and in eukaryotes there are many replicons per a single DNA molecule. Okay. Now, here you can see there is a single replicon, that means the total DNA is a single replicon or single replicating unit. Once it is being initiated means it will go on synthesizing the whole length of DNA or replicating the whole length of DNA into two such double stranded DNA. But in case of eukaryotes, now as we know the nature of the DNA, these are linear. Okay. Now, there are many such replicons, they may be overlapped like this. Okay. So, like this. So, this is one replicon, this is one another, this is another, this is another. There may be 2, 3, 4, but there will be many. There will be many in nature. Okay. So, as we can see here, now these are all replicon. Now, this A is a prokaryotic replicon. Okay. Now, you see this is a single DNA double stranded DNA molecule. Now, here this from this point, this is the origin of replication. From this point it goes on synthesizing now into two double stranded molecules. But in case of eukaryotes, now as you can see, now these are all replicons. Now, or the origin of replicants. Now, from here, now they will go on what? Synthesizing the double stranded DNA or 2 strands of DNA like this in many replicating units. Okay. Now, this is not that this is a single replicating unit, rather you can see, now here you can see 5 replicating units are there. Okay. It will go on replicating 2 double stranded DNA, but they will also have 5 such replicating units or replicons. Okay. So, what a replicon is? A replicon is a segment of DNA, okay, is a segment of DNA that can replicate itself, well, that can replicate itself independently. That means let us suppose, now here how many replicons are there? 1, 2, 3, 4, 5 and 6, okay. Now all the 6 will not replicate at one time. This may replicate at one time, this may replicate sometimes later, okay. So togetherly within the S phase they will replicate as per their convenience, okay. So a replicon is a unit of DNA segment, or this is a segment of DNA that is capable of what? Replicating itself independently, okay. So that is why the prokaryotes have a single replicon, whereas the eukaryotes has many replicons. Now a replicon has a single, it must have a single unit of replication, or it must have a single origin of replication, okay. So a replicon always have a single origin of replication, okay. So what is origin of replication? Now here you see that this point, now DNA replication, it won't initiate, or it does not initiate from any point it likes, okay. So there is a specific point from which your DNA replication starts, that is a specific point. So what is that specific point? That specific point is known as origin of replication. Now let us suppose if this is the what, this is the double stranded DNA, or this is the whole length of DNA that has to be replicated. Now it does not mean that it will start replicating from this point or this point or this point, this is not that, rather there is a fixed point, okay, which is called as origin of replication. Well, so let me draw it here, it would be better. Now let us suppose this is a prokaryotic DNA strand. Now there will be a single point, this point, and from this point it will go on synthesizing itself like this, okay. So this point is called as origin of replication, okay, and from this point the DNA will go on synthesizing like this, completing the whole unit. So replicon and origin of replications are very important. Replicon is the unit of replication, and origin of replication is the point from which your replication initiates or starts. Replication does not initiate at any point it likes, rather there is a fixed point from which your replication initiates, and that fixed point is called as origin of replication. So let us have a look on the origin of replication of E. coli. Now ORIC is the origin of replication, of the bacteria called as E. coli, okay, E. coli. Now you see this is the total E. coli genome, that is this is the double stranded circular DNA, and this is the point known as ORIC or origin of replication of E. coli, from which your DNA replication will initiate, and it will go on synthesizing the whole strand into 2 DNA molecules, okay. Now this ORIC is a 245 base pair long DNA strand. This is this ORIC that I showed you here, this point, okay. If this is the ORIC point, this is actually, as you can see, now this is a long, this much length is that, and this length is 245 base pair long, Okay. Now, what features it has? It has 313 base pair repeats which are 80 rich in nature, and 9 base pair repeats, 4 9 base pair repeats which may be GC rich or whatever, but this point is important, this 313 base pair repeats which are 80 rich in nature. Now, what does it mean by 80 rich? 80 rich means now when a DNA, now you know that the two strands are complementary to each other. The complementary basis forms the hydrogen bonding which makes the helical structure of DNA. Now, if the hydrogen bond is broken, okay, now it will create two DNA single stranded template. So, at which point the hydrogen bond has to be broken, where it has to be weak or where it is weak, then at that point it has to break. So, the breaking point should be 80 rich in nature. Why? Because there are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. So, which will be much easier to break? Obviously, the 80 bonds. Okay. So, that is why this 313 base pair repeats are 80 rich in nature. So, what happens? This ORIC DNA wraps around a molecule known as DNA A complex. Okay. This is a monomeric molecule. So, it wraps like this. Well, now these are the 13 base pair repeats. So, you see this is the 13 base pair repeat. Now, here this wrapping denatures, because of this wrapping, this point, this 13 base pair repeats, now this will be what? This will be denatured and there will be formation of 2 single stranded template. Now, you see this is double stranded in nature, and here the double strand nature will be what? Will be converted into 2 single stranded template, and these 2 single stranded templates are now exposed, or these are now ready for the synthesis of 2 new strands of DNA by the help of the replisome system or the replisome complex. For example, it may be held by helicase enzyme, topoisomerase enzyme, primase, DNA ligase, your DNA polymerase, the most important enzyme. Okay. And finally, there will be, after this one, after the opening of the double stranded DNA into 2 single stranded template, now there will be loading of the DNA B to DNA C complex. Okay. That is nothing but the helicase. Now, you can see the helicase in this violet colors, and some this yellow colored portions, or these are called as, these yellow proteins, these are called as single stranded binding proteins, which binds to the single stranded template in order to prevent further hydrogen bond formation between the complementary bases. Okay. So, this is ORIC, the origin of replication of E. coli. So as the other origin of replication having different kind of features, but this is the feature of the ORIC. Let us see now how the replication is initiated. So, once again, now within this, now this is the origin of replication. So, this is the origin of replication. Okay. At this point, lots of DNA A molecules bind, or this DNA or this ORIC portion, it wraps over the DNA A molecules. Okay. So, after the wrapping, it will generate 2 single stranded DNA template. Okay. 2 single stranded DNA template. Now, next what happens? Now, next there will be loading of DNA what? Helicase, or simply there will be loading of helicase protein or helicase enzyme. Okay. So, this helicase enzyme in case of prokaryote is DNA B protein. Okay. But this forms a complex with DNA C protein and it forms a hexameric structure like this. For example, if this is the DNA B, now along with that 6 molecules of DNA C forming a hexameric, okay. Now this will be loaded to this what? To this single stranded region, okay. If it is bidirectional, 2 helices, so as the nature of this DNA replication is bidirectional, DNA replication can be unidirectional, it can be bidirectional. If it is bidirectional, now 2 helicase proteins or helicase complex will be loaded at these 2 points, okay. And after the loading of helicase, now because of the generation of the single stranded DNA, now the single stranded binding proteins, they will bind to these portions, so that it will prevent further formation of hydrogen bonds between the complementary base pair. So what happens? Now the single stranded templates are intact for the synthesis of new strand, okay, and these 2 strands will join together. So this is the initiation of replication. So what happens? The initiation starts with the wrapping of the origin of replication over the DNA molecules. It will create or it will break the bond between the AT rich regions, and it will create 2 single stranded template, and this 2 single stranded template will be loaded with a helicase B complex, okay, and this helicase complex will go on, okay, widening that single stranded region, and it will come in the form of a replication eye, replication bubble or replication fork, okay. So in the next slide we will see the unwinding of DNA strand at the origin of replication. So how it happens? You see, this happens by DNA B or by a helicase enzyme, okay. Now what are helicase enzyme? Now once the single stranded nature, once it will be created, now the helicase enzyme will be loaded to that point. Now it will go on cutting, okay, the hydrogen. Now these are the hydrogen bonds, so it will go on cutting these hydrogen bonds and making single stranded template, okay. So this is an enzyme, or helicase is an enzyme that unwinds the double helix by breaking the hydrogen bonds between the complementary bases. You can see it like this. So after some time you will see one strand as this and another strand as this. So 2 single stranded DNA templates will be created. After that, now RNA primase will be loaded, it will form a RNA primer, okay. Then after that it will synthesize a short RNA primer which will provide a 3' hydroxylic group, and after that the DNA polymerase will be loaded and it will go on synthesizing new strand basing upon the template strand, and it will pull the complementary bases from the nucleoplasmic pool, and as the complementary bases will come, automatically it will create the hydrogen bonds between the template complementary base and or the template nitrogenous base and the new strand that has to be synthesized, that's nitrogenous base, okay. So the DNA helicase enzyme, its role is to cut the double stranded DNA into single stranded template. Now, so here it creates a replication fork. So if this is the point of origin of replication, if this is origin of replication, now from this point, now the helicase enzyme, this will go on, as I showed you, from this point and this point, as it is bidirectional. Here the replication is bidirectional, okay. So it will go on what? Opening the double stranded. Now you see here these are double stranded, okay. So it will go on widening the single stranded region like this, and the double stranded region will be converted more and more to single stranded region, so that new strand will be synthesized basing upon the old strand. So, what it is? This portion is the what? Replication fork, this total portion, this is the replication fork, and this replication fork moves towards this direction and this direction, as it is bidirectional DNA replication. If it would be unidirectional, then it will move to a single direction. If it is bidirectional, it will move in both the direction. And here, now what will be loaded here? Now helicase protein will be, or helicase complex will be loaded here. Okay, one at this point, another at this point. Okay, and it will go on widening the single stranded region and basing upon which new strand will go on synthesizing. Well, but now you see how the fork is moving. Now this is for an eukaryotic chromosome or for a eukaryotic DNA. Now as you can see, now there are many origin of replication or initiation of replication. So as there are many origin of replication, now here you can see many forks are seen, and these are called as what? Replication fork, replication fork or replication eye or replication what? Bubble. So as it looks like an eye or a bubble in the form of a bubble, so that's why these are called as replication fork, and the fork moves in opposite direction. Here one direction, here another, from there also in this two direction. Now what will happen? It will go on synthesizing like this, and at one point they will meet where both of the strands would have been synthesized. Okay, so this is called as the fork movement. So before going to that, let me tell you the function of the DNA helicase. Topoisomer is another one enzyme and all. Now let us see. Now this is the helicase protein. It will go on cutting the hydrogen bonds, generating single stranded template. Once the single stranded templates are generated, they will be bind by what? Single stranded binding proteins. Well, but one thing is that as the DNA is very much supercoiled, okay, so the cutting of the DNA helicase or the cutting of the double stranded of DNA by DNA helicase into single strands creates a torsional strain, or it creates somewhat of stress in the DNA molecule. Okay, that stress or strain, it is relieved by another enzyme. Well, that is called as topoisomerase. So this stress, that means the double stranded nature, it is being converted into single stranded nature. As it will go on cutting more and more, it will create a negative impact on the double stranded DNA molecule. So it will be more coiled, or somewhat this will impose a strain on that, and what happens? In order to relieve that strain, the topoisomerase or the DNA gyrase enzyme, same is the thing. So the DNA gyrase in case of prokaryotes, now the DNA gyrase enzyme, it comes into action. So what it will do, in order to create any kind of disorganized structure in the double stranded DNA, now it will create a nick. Okay, so it will create a nick in the double stranded and it will make it straight, so that there will be no hindrance in what? No hindrance for cutting of the double stranded DNA into single stranded template by the helicase enzyme, and the topoisomerase will go on converting the strain, or it will go on releasing the strain or the stress in the DNA molecule. How? By nicking the DNA molecule and again sealing it. Okay, so that is why the topoisomerase enzymes are called as nicking and relaxing enzymes. Okay, so it relaxes the DNA molecule for the act of DNA replication. So first is helicase, then is topoisomerase, then single stranded binding protein. These three we have completed. Now next, as the DNA strands are generated, single strand DNA molecules are generated. Now what happens? Now here in this case, now another molecule comes into picture, that is the primase molecule. this prime is nothing but a RNA primase. So, what it does, it synthesizes a short strand. It synthesizes a short strand of RNA molecule, short strand of RNA of 10 to 20 nucleotides long. Okay, 10 to 20 nucleotides long. Okay, so here you see it creates a RNA primer. Now that RNA primer, what is the role of that RNA primer? That RNA primer, if this is the primer of RNA, it exposes a 3' hydroxylic group, okay, a 3' hydroxylic group, as you can see. So it exposes a 3' hydroxylic group here, so that now next the DNA polymerase enzyme will come and will bind here, and it will go on adding the nitrogenous bases one after another and forming the hydrogen bonds. So it will go on forming the hydrogen bonds. Okay. So why a primer is formed? Because DNA polymerase enzyme, it cannot synthesizes the new strand of DNA de novo, as it requires a free 3' hydroxylic group, and that 3' hydroxylic group is being provided by a short RNA primer, which exposes a 3' hydroxylic group to the enzyme, that is DNA polymerase. Okay, DNA polymerase. So this DNA polymerase enzyme, then it will go on synthesizing what? This strand of DNA continuously like this. And the helicase enzyme also, it goes on what? Breaking the hydrogen bonds and creating the single stranded template, as we can see it here. Okay. So in this way there will be RNA priming. So the formation of the RNA primer is called as RNA priming, and the primosome is formed by the assistance of this helicase topoisomerase and the primase enzyme. Okay. So this is the complete picture where we can see all the enzymes and proteins that are involved in what? In DNA replication. Now next, now as you can see, now after the formation of RNA primer, there will be loading of DNA polymerase enzyme, and the DNA polymerase enzyme, now the DNA polymerase enzyme has two parts. One part will go on synthesizing. Now here you can see the green in color, this is the RNA primer. So once the primer is formed, the DNA polymerase enzyme, it will go on synthesizing the DNA strand, okay, continuously, as I told you previously. So it will go continuously. So it will go on synthesizing continuously in this direction a strand which is called as leading strand. Okay. Why it is called as leading strand? As it is synthesized continuously, that is why it is called as leading strand. But what happens to the other strand? The other strand, as you know, the direction of synthesis, it is from 5' to 3' direction. So here you see this is from 5' to 3' direction. Okay. Now when in the other strand, it also has to keep the same direction. So from 5' to 3'. So what happens? Now here also RNA priming occurs, but it occurs many times as the fragments are synthesized. Okay. So that is why one strand is called as leading strand, the strand that is synthesized continuously by the formation of a single RNA primer, whereas the other strand is called as lagging strand, as it is synthesized in fragments. Okay. As it is synthesized in fragments, and these are called as Okazaki fragments from the 5' direction to the 3' direction, but many primers are formed, many RNA primers are formed as many Okazaki fragments are formed. And as this strand is synthesized in patches, okay, like this, if I will extend here, this will also come in form of what? Patch like this, okay. So, these are called as Okazaki fragments as per the name of the scientist, and this is the fork progression, and in this way the leading strand is synthesized continuously, the lagging strand is synthesized discontinuously, okay. So, this is synthesized discontinuously. So, this is about, then what happens towards the last? Now, after you see this is bidirectional, so this is the origin of replication, this is bidirectional DNA replication. Now, what happens here? A single primer is formed in this leading strand, this is the leading strand, and here many RNA primers are formed. So, what happens? All the RNA primers will dissociate, or they will be taken out of this DNA strand, and instead of that another DNA polymerase enzyme will go on what? Removing this strand, and instead of it synthesizes a short DNA strand, and it will be what? They will be joined together by the help of the enzyme called as DNA ligase. So, DNA ligase helps in ligating all the DNA molecules that are present on the lagging strand synthesis. So, this is all about your DNA replication. Thank you so much.